The diversity of life on Earth appears entirely chaotic. An animal can be a stationary filter, a gelatinous bell, an armored scavenger, or high-speed predator. When scientists catalog this variety, the numbers are staggering. We have described over 1 million distinct species in the animal kingdom alone. Faced with that volume, the standard approach is often rote memorization, trying to force 1 million isolated facts, phyla names, and body traits into your head. That strategy guarantees failure. Looking at this textbook page and the hand-drawn classification notes, we can see a much better method. Biological classification is not a guessing game. It is a rigorous, predictive framework. True taxonomy is an elegant sorting algorithm driven by deep, structural rules that govern how life is built. An organism's final, complex structure is entirely dictated by its foundational architecture. To sort every animal on Earth, we only need to define 4 core structural parameters: levels of organization, their body symmetry, the number of germ layers, and the nature of the coelom. These parameters are established at the very beginning of life, in the earliest stages of embryonic development. After fertilization, as a single cell rapidly scales into a hollow sphere of thousands, these architectural decisions are locked into place. These 4 physical rules allow us to derive the animal tree of life from basic first principles. The logic begins at the lowest organizational tier, the cellular level. Here, cells act as loose aggregates rather than a unified structure. The first major upgrade in biological hardware is diploblastic organization. Cells arrange themselves into 2 distinct functional layers, an inner tissue and an outer tissue. This cross-section shows the diploblastic state perfectly. You have the outer ectoderm in blue and the inner endoderm in red, separated only by an undifferentiated, jelly-like layer called the mesoglia. The most crucial evolutionary leap in tissue organization happens next, the transition to a triploblastic body plan. Panning to the next diagram, we see the specific addition that defines this tier. A distinct, structured third layer emerges right in the middle, the mesoderm. The physiology of the animal is permanently upgraded. The mesoderm is the absolute biological prerequisite for building complex internal structures, like true musculature and cardiovascular systems. This middle tissue layer provides the structural material required for complex internal organ systems. Moving from internal tissues to external geometry, the layout of the organism provides our next sorting parameter. Radial symmetry is a structural plan where the body revolves uniformly around a central axis, like spokes on a wheel. Looking at this diagram of a starfish, you can run a plane through the center at multiple angles and consistently divide the animal into identical halves. This design is biologically optimized for organisms that are passive or anchored in place, allowing them to interact with their environment equally from all 360 degrees. Bilateral symmetry provides a stark contrast. In this design, the body can only be divided down a single plane into distinct left and right halves. Dropping down to the crab diagram, you see there is only one central vertical axis that creates identical halves. This single-axis geometry provides the essential streamlining and precise muscle distribution that is absolutely required for rapid, directional movement. A bilateral body plan is built specifically to cut efficiently through water or propel across land. The physics of moving forward creates a new necessity. Your sensory equipment must lead the way. This constraint drives cephalization, the heavy concentration of nervous tissue at the anterior end, creating a distinct head. Body symmetry serves as the fundamental blueprint for how an animal negotiates the physical forces of its environment. The third and heavily tested architectural rule determines the nature of the body cavity, known as the coelom. In the primitive acoelomate state, the internal space situated between the gut tube and the outer body wall is packed entirely solid. This cross-section at the bottom shows the acoelomate structure. The tissue layers sit flush against each other without a single internal gap. In contrast, a true coelom is a distinct, fluid-filled space that is perfectly lined by that critical mesoderm tissue. Panning up to the top diagram, those bright open cavities, wrapped completely inside and out by the red mesoderm, represent the true coelom. Mechanically, this fluid chamber serves as a biological shock absorber and provides a hydrostatic skeleton for muscles to push against during locomotion. Most importantly, the true coelom provides the empty physical space required for complex organs to grow, coil, and operate entirely independently of the outer body wall. Without a true coelom, a stomach cannot churn, a heart cannot beat, and lungs cannot expand. Biology is driven by trial and error, and there is one structural middle ground: organisms that attempt to build a body cavity but fail to fully line it with mesoderm. This is known as the pseudocoelomate, or false cavity, condition. Look closely at the middle diagram. The mesoderm exists, but it's broken up into scattered, disconnected pouches. It never forms a continuous protective lining. The Aschelminthes, or roundworms, are the specific phylum that uniquely occupies this transitional anatomical state. Observing this partial step proves that the taxonomic tree maps literal physical progression. We can now take these individual architectural rules and run the actual classification algorithm. This master flowchart categorizes the entirety of kingdom Animalia based entirely on the parameters we just defined. The very first logical split accurately isolates the porifera, the sponges, purely because they never evolve past that basic cellular level of organization. If an animal develops tissues but retains radial symmetry, the algorithm tracks downward to Quilenterata and Coniferea. Following the bilateral branch reveals the final split. The coelom's nature takes the remaining phyla and flawlessly sorts them into position. Applied sequentially, taxonomy becomes a predictable equation. An organism's position on this logic map predicts its specific physiological systems. We can translate this sorting logic directly into a final data matrix. This summary table maps out the major phyla against their highly specialized organ systems. Notice the cascading diagonal pattern of complexity running from top left to bottom right. Track the digestive system column as it shifts predictably from absent to incomplete to definitively complete. A complete digestive system mathematically locks into place only after the organism secures both bilateral symmetry and triple blastic layering. Look at the circulatory system column on the right. Its presence is strictly restricted to the most advanced true coelomates at the bottom of the board. Once you establish an organism's foundational architecture, predicting its internal systems is a guaranteed debection. Every complex animal reflects the foundational cell it grew from. Modern taxonomy serves as a map of reverse-engineered evolution, built from the bottom up using these fundamental first principles.